Hello, today I will be talking about nitrogen fertilization of tomatoes in an elevated carbon dioxide world. And I know that people aren't really working with tomatoes mostly in this conference, so also the possible ramifications for other C3 plants such as fruit trees. Sometimes I read older articles. This one's from 1995. And one of the things that's interesting about reading older articles is whenever they talk about ambient carbon dioxide, you see that it's totally different than it is today. If you look at the bottom of this screen over here, you see that the ambient carbon dioxide from this 1995 paper is 350 ppm. I've been writing a review also on the effects of elevated carbon dioxide on plants. And in my introduction, I wrote over a year ago, and I had the ambient carbon dioxide at 413 ppm. And then I'm about to publish it, and I decided to update it, and by now it's 416 ppm. So if it takes you more than a year to write a review, then you need to change the ambient CO2. There are direct and indirect effects of elevated carbon dioxide on plants. The indirect ones have to do with climate change and drought and heat and sea level rise and all sorts of things. But we're not going to talk about those today. We're talking about the direct effects of elevated carbon dioxide on plants. I know that most people are working on fruit trees. I worked on tomatoes. In the format of this chart presentation, we're going to have a demonstration of the effects of elevated carbon dioxide on C3 plants reflected through our work with tomatoes. A discussion of the mechanisms leading to some of these effects, again, reflected through our work on tomatoes. We're doing tomatoes not because we're so interested in tomatoes, but mostly because we're using it as a model plant for all plants. And the last thing we're going to do is a discussion of the amelioration of some of these effects reflected through, you guessed it, our work with tomatoes. But before we get to tomatoes, I actually did do a little bit of work on lemon trees, which is a little bit closer to the work done in this conference. And one of the effects of elevated carbon dioxide on every C3 plant, but also on lemon trees, is that the higher the carbon dioxide level, the less transpiration there is. In this experiment, we had 60 lemon trees growing in 3 different levels of carbon dioxide. And over the course of 24 hours, we weighed the lemon trees. And we saw that the ones grown in 400 ppm carbon dioxide transpired more than the ones that were grown in 850 ppm, about 20% difference. All right, now to the tomatoes. Our experimental setup, we have 2 long-term experiments. The time frame was 3 months. The variables that we changed were 5 levels of nitrogen, 10, 30, 50, 70, and 90 ppm in the irrigation water. It was grown in perlite. And 3 levels of carbon dioxide, 400, 750, and 900, and 6 repetitions. And things that we measured in these experiments were things that changed over the long term because they were our long-term experiments. So we looked at the NPK concentration in the leaves, the biomass and nitrate concentration in the leaves. We also did 2 short-term experiments. Each one was 1 week. In these, we only had 2 levels of carbon dioxide and 2 levels of nitrogen. The measurements that we took were the gas exchange measurements. We did photosynthesis, conductivity, ACI curves, Rubisco expression, and leaf starch concentration. First thing that we see, and everyone sees whenever they grow things in elevated carbon dioxide, is an increase of biomass. If you look over here, this is, I thought I had this on here, but the black is the low carbon dioxide, and as it gets lighter, it gets to higher carbon dioxide. And the more carbon dioxide you add, the bigger the plant gets. This is the biomass. And you could also have an interactive effect here that you can see that the higher levels of nitrogen, there's more of an effect of the carbon dioxide. See, at 90, it jumps, whereas at 10, it kind of stumbles a little bit up. And there wasn't a statistically significant interactive effect, but this is actually what a lot of papers see. They don't see a statistically significant effect, but they see a trend. And when you do meta-analyses of how carbon dioxide affects biomass, you tend to find that nitrogen does affect how much the carbon dioxide impacts the biomass. Another thing that we always see is that the leaf starch goes up. The more nitrogen, the more carbon dioxide in the air, the more starch there is in the leaf. You see here the high nitrogen and the low nitrogen levels, and each one has a low carbon dioxide and high carbon dioxide. And what we see is that the higher carbon dioxide levels have more starch in the leaves. I think this shows that there's a buildup of starch. It means that there's a sink source imbalance here, that plant is making more starch than it knows what to do with, and it kind of builds up in the leaves. You notice that it happens more in the low nitrogen than in the high nitrogen. But it happens in both, it happens in both of them. But in the end, the absolute value of starch is always higher in the low nitrogen than in the high nitrogen, even if you add in the carbon dioxide. So does increased N ameliorate starch buildup? Well, not exactly, because there's more starch in the high carbon dioxide no matter what, but it counteracts it, meaning if you have more nitrogen, there's always going to be less starch. Another thing we're going to talk about is photosynthetic acclimation to elevated carbon dioxide. Before we get into it, we're going to do some theory. What happens if you take two identical plants and put one of them in elevated carbon dioxide? As we saw before, one of them grows bigger. Why does it grow bigger? Because there's more photosynthesis. Why is there more photosynthesis? Well, carbon dioxide is the substrate for photosynthesis. But what happens if you then take that second plant, both plants again, and you put them back into 400 ppm carbon dioxide? You see that the photosynthetic rate of the larger plant, the one that had been growing in 850, now is lower than the one that was used to growing at 400 all this time. So which one has a higher photosynthetic potential? The one that has been growing in 400. There are two sort of types of photosynthetic acclimation. They both occur at the same time, but there's two different processes. One is there's stomatal and non-stomatal. Stomatal is actually just the stomata in elevated carbon dioxide closed. That's why the transpiration was lower in the lemon trees that were grown in elevated carbon dioxide. The stomata are closing. And as the stomata close, less carbon dioxide goes into the leaf, and that could lead to some sort of photosynthetic acclimation. The other level of acclimation is that the photosynthetic machinery itself inside the leaf gets downregulated. Researchers mostly focus on the rubisco. There's less rubisco in the leaf of plants grown in elevated carbon dioxide compared to those grown in ambient carbon dioxide. How do you measure photosynthetic acclimation? Well, we measure it. One way to measure it is you put them both in a room with 400 ppm, and then you measure the photosynthesis at a standard CO2, and you can see that the one that's used to the higher carbon dioxide has a lower level of photosynthesis. You can also measure the concentration or activity of photosynthetic enzymes, such as rubisco, or the expression of the genes of the photosynthetic enzymes. You can also measure the photosynthesis at multiple carbon dioxide levels and plot an ACI plot. A is the photosynthesis, and CI is the level of carbon dioxide within the leaf next to in the cell itself. And you can compare the slope of the graph at different points. And that will tell us VC max is the slope at the beginning where the carbon dioxide is low, and J max is the slope where the carbon dioxide is relatively high. And you see here the limiting, VC max basically tells you the activity of the rubisco, and J max tells you the activity of the electron transport system in the leaf. The importance of photosynthetic acclimation. Well, mostly one of the importances is it's a missed opportunity, right? Because the plant, it has higher photosynthesis than it would have had you had it in 400, but it still isn't as high as it could be, because if you didn't have this acclimation where the rubisco goes down and the stomata close, you could have an even bigger plant. And with the population of the world going up and up and up all the time, it's expected to be at 9.8 billion, I think, in 2050. It's important that we get all the calories we can from the sun into our plants. Okay, so let's talk about photosynthetic acclimation in my tomato plants themselves. if you look at the net photosynthetic rate, this is measured at 400 ppm, you'll see that those plants that were grown in ambient carbon dioxide do better or have higher levels of photosynthesis than those that were grown in lower levels of carbon dioxide. Sorry, than those that were grown in higher levels of carbon dioxide. You'll notice though that the nitrogen, if you add nitrogen, if you go from high nitrogen to low nitrogen, sorry, low nitrogen to high nitrogen, it doesn't ameliorate it, because either way, the photosynthetic acclimation seems to occur. It occurs both in the high and the low nitrogen. However, if you look at the absolute value of the photosynthesis is lower in those plants that were grown in elevated carbon dioxide, but it's higher in those plants that were given high nitrogen. So the high nitrogen somewhat counteracts the effect of the carbon dioxide. Even if both of them end up affecting the plants, they affect them in different directions. You can also look at the stomatal conductance, as we saw in the lemon plants, lemon trees. In tomatoes, it happens too, right? You see that the stomatal conductance goes down. The stomata have been closing because of elevated carbon dioxide. And you can look at the Rubisco expression, and you can see that the Rubisco expression falls when you grow something in elevated carbon dioxide. You can also look at the VC max, which we said was could be derived from the ACI curve, and it expresses the Rubisco activity, and you can see that it goes down up here. It goes down when you go from high carbon dioxide to low, when it go from low carbon dioxide to high carbon dioxide. On the other hand, again, if you look at how nitrogen fertilization changes the level of I guess the absolute value of the VC max, you see that you can grow the VC max by adding nitrogen. I didn't work with any of these other things, but photosynthetic acclimation occurs also in trees and legumes and other plants as well. This is a meta-analysis by Elizabeth Ainsworth from 2007, and she found that different plants are affected in different amounts. This is the VC max that we were talking about before, and how it's affected by elevated carbon dioxide. And she saw basically that legumes are affected the least. Legumes and trees are affected the least, at around -6 or -7% response of their VC max when you grow in elevated carbon dioxide, and crops have more. So my tomato plants, which is -17%, that's pretty close to what I got if you go backwards. VC max, it goes from, like 50 to 40, that's 20% difference. And here it's a 17% difference. Another meta-analysis of trees. It's not fruit trees, it's forest trees. But you get the same idea that if you check the photosynthetic rate at 350, again, this was it, 1999, so they were still at around 350, I guess, or they used 350 as their standard. You get an effect size of basically -19% in your photosynthesis in plants grown in elevated carbon dioxide and then measured in 350. Hypothesis about the causes of photosynthetic acclimation. There seems to be caused by a sink source imbalance. When you have more carbon dioxide, it has more photosynthesis, and then with more photosynthesis, you get more starch and more sugars, and the plant doesn't seem to be able to balance that source with a sink. It doesn't know where to put all that those photosynthates. And there's a sugar sensing enzyme called hexokinase, which tells the plant how much sugar or extra sugar they have. And then there's a downregulation of the Rubisco gene, mostly the small subunit of the Rubisco gene. This is the mechanism that was put forward by Moore et al. In 1999, and it's basically, they still talk about it today. Evidence that we have to show this process, well, we didn't show the whole process, but as we saw in our previous slides, we did have a decrease in the Rubisco expression of the small subunit, and we did have an increase of starch, showing that there was a sink source imbalance. Okay, next thing I want to talk about is elevated CO2 induced nitrogen deficiency. If you look at the top graph over here, whenever you grow plants in elevated carbon dioxide, here are my tomato plants, you end up with leaves that have less nitrogen in them. Their percentage of nitrogen within the leaves is smaller. Again, we can look at how if increasing of nitrogen ameliorates this. And it doesn't exactly, because in the highest nitrogen level, you still have this drop here. Whenever you add carbon dioxide, you end up with a drop of nitrogen. But on the other hand, as we said before, the absolute value of the nitrogen is higher in the 90, even in the 90 ppm nitrogen, even in the higher levels of carbon dioxide. So the absolute value of nitrogen concentration is increased by elevated carbon dioxide. The importance of ESIND, first of all, human health. When you end up with plants that have less nitrogen in them, you have plants that have less protein in them. Animal health, if the animals are eating the leaves. Insect health, if the insects are eating the leaves. And pollinator health, if the pollinators are eating the pollen, which ends up having less nitrogen in it. Mechanism causing ESIND, you can have dilution and uptake inhibition. Nitrogen uptake could be caused by the limited transpiration, perhaps, as there's less mass flow, or nitrogen assimilation inhibition with limited reductants. Because when the plant is reducing the carbon dioxide to starch, it has less reductant available to reduce nitrate to amino acids. Can elevated carbon dioxide induce nitrogen deficiency be attributed entirely to dilution? Often in the literature it can't be, but in this one it seems to be able to. That in our work, you can see that if you look at the total leaf N, not the nitrogen concentration, you see that it is the same. Basically, the plant took up the same amount of nitrogen, but in the end, its concentration is less. Do we have a trouble with nitrate assimilation or with nitrate uptake? Well, if you look at the amount of nitrate in the leaves, you see that the more carbon dioxide you have, the less nitrate you have, which seems to indicate that the problem is with uptake and not with assimilation, because assimilation should take away the nitrate and uptake adds to the nitrate. And here we see that the higher the carbon dioxide, the less nitrate is sitting there in the leaf unassimilated. How do you ameliorate photosynthetic acclimation and ESIND and the nitrogen deficiency? From our work with tomatoes, you can see that changing the nitrogen concentration can counteract, if not ameliorate, the effects of the elevated carbon dioxide. Conclusion: in the future, it may be worthwhile to change nitrogen fertilization of tomatoes to get the most benefit from the elevated carbon dioxide and to counteract some of the negative effects of elevated carbon dioxide. In fruit trees, which are also C3 plants, probably will require some modification of nitrogen fertilization as well. Thank you.